Uh, Today, I am going to present the topic on gene regulatory network, which is purely based on mathematical modeling and simulation of gene regulatory network. Here, basically, I'm going to show in the form of a kind of a small master thesis presentation. So the title, which is Simulated Evolution of Gene Regulatory Networks, that means here we have a small system of gene, that is a network of some certain number of genes, and then evolution simulated, that is via simulation. So basically this is a kind of understanding. Now the overview of gene regulatory networks. Here what I am going to cover is basics of gene regulatory network, methods used. Normally, there are two common methods which are used, like continuous and discrete. And then third thing is the algorithm by which we are going to evolve the system or optimize the parameters, where we are going to check the effect of mutations. And there are two kinds of mutations which in this model we have covered. One is environmental effect, the mutation as a result of environmental effect. Another could be, let's say, genetic or epigenetic, genomic or epigenomic. So the continuous model is like from the name, it is clear that continuous model, nothing break up in between, continued simulations. And then discrete is like, as it is clear from the name, it is discrete means Boolean here. So the algorithm which we have used is evolutionary algorithm. And then next part will be the results, outcomes which we are getting after setting up the model and evolving them under certain conditions. And finally, the summary. So now, the basic is gene regulated networks are collections of genes in the form of like connectivity, connected with certain genes or all the genes in a directional way or non-directional way. So the connectivity could be directional also. Directional means activation, deactivation, non-directional. That means we just know they are connected with each other, but we don't know what is the nature of connection. For example, gene 1 is connected with gene 2, and if it is directionless, that means we don't know if gene 1 is activating gene 2 or gene 2 is activating gene 1 or inactivating. And in direction, it is fixed, decided if it is one of them is activating and another is deactivating, or maybe one is activating. That is the only one connectivity possibility. So here, connectivity represents the interaction to modify or change the other's behavior. That means these connectivities are or will be the decision factor for the cells to make any decision or biological functions. So here it is also clear why we are interested in gene regulatory networks and how much important it could be. Sometimes it is also possible that small sets of genes in a big gene network are acting as a main decision making entity, and that we call it as subnetwork or motif, and that we also called as building blocks of the network, that is network motif or subnetwork or subgraph. Small modifications to a gene regulatory network could lead to a big functional or phenotypic changes, such as transient perturbations. Transient means short term change in of a gene's activity could force a cell to differentiate or could force a cell to go for apoptosis or proliferation or growth. So this is quite basics to understand. Now, gene networks, regulatory networks, which I have used here are random networks. That means the connectivities are not decided. So in general, there are three types of networks. One is called scale-free, another is called hierarchical, third one is random. So here I am going to